So now we have numerical data. Notice that I circled a whole bunch of numbers that were really high, like 18.9, 18.1. That means that those values are very different because it's looking at differences in mitochondria. I will tell you many, many times students make like some really just simple mistakes because they look at those numbers and they assume that it might be a similarity, but this is looking at divergence or differences. So as you go through some of these cladograms, just take that extra minute to see, are we looking at similarities or are we looking at differences? And this is looking at differences. So high numbers are going to show, you know, a more distant evolutionary history because it's going to have more differences. Notice that all of those numbers in blue are associated with comparisons to the gibbon. So the gibbon is the most dissimilar in its mitochondrial DNA compared to the other primate species. Because of that, we know it's in the outgroup. The outgroup is going to always be the most dissimilar organism. So down here at the bottom, that's the common ancestor. And here's the first outgroup right here. That's going to be the gibbon. AP gave you the outgroup. Thanks a lot, AP. The outgroup's the most easiest thing to determine, so thanks for nothing. Like that's sometimes frustrating because usually finding the outgroup is the easiest thing. But if they didn't give you the gibbon, that's how we determine what the outgroup is. It's the one with the most differences. Next, we have to determine this sequence, all right? Like how did I determine human, chimpanzee, gorilla, and orangutan? That's going to be a little bit more challenging. So let's first start now with looking at the number that I circled in red. I always find the outgroup first. Which one has the most things that are dissimilar? And that's easy to determine, that's given. But now I circled that 8.8 in red, and that's showing that these two species are the most closely related. They have the most similarities. So the human and the chimpanzee, closely related because there's not very many differences in the mitochondrial DNA. So notice that I put them very close together on the cladogram. What I want to let you know is that, see here where I said this is interchangeable? I put the human on top. That doesn't mean that humans, you know, are necessarily like more evolved or better than a chimpanzee. Like every organism is well evolved to its environment. But it's interchangeable because this node right here, where those two lines diverge, that's called a node, and it represents an extinct common ancestor to both the human and the chimpanzee. If that is like the point in the path and you could go right or you could go left, doesn't mean that like you couldn't interchange those and those would mean the same thing. So the chimpanzee could have been at the top of the cladogram and the human could have been at the next line too. Those would both be correct because anything around a node could be flipped and it's still showing the same relationship. So we determine the outgroup by the one that has the most differences, and we can determine which organisms should be closely aligned to one another based on their similarities. And now we have to determine, like, how do we know that the gorilla is going to be closer to the human and the chimpanzee and not the orangutan? So let's go back up here a little further, and we're going to look back at the data. I'm going to put this in green. And notice here that if I look at the human compared to the gorilla, that's 10.3. And if I look at the gorilla compared to the chimpanzee, that's 10.6. That is a smaller number. That's going to show more similarities than the human compared to the orangutan, that's 16.1, or the chimpanzee compared to the orangutan, that's 17.2. So I know that that orangutan should be further away from the human and the chimpanzee than the gorilla, and the gorilla should be closer to both of those organisms. So that's how I showed it on the cladogram. The gorilla should have a closer relationship to the chimpanzee and the human, and the orangutan will have a more distant relationship to those primates. How about any questions for me on how we determine that sequence on the cladogram? Okay. Hopefully we're feeling a little bit better with that. Let's move on and talk about number 5. And on number 5, it says, on the cladogram, draw a circle around all the species that were descended from the species indicated by the node in the square. So we just talked about what a node is. And it's talking about this node right here. And that node is showing an extinct common ancestor. And notice when we go through that node, I can go through the node to get to the gorilla. So that's a common ancestor to the gorilla. But I could also go from that node up to the human and over to the chimpanzee. So that would show a common ancestor to all 3 of those primates. Obviously, if I look at the node between the human and the chimpanzee, that's a more recent common ancestor. This node is a more distant one, but this node would be a common ancestor of those 3 organisms. And then finally, the last question here, number 6, is to provide reasoning for the placement of the gibbons. Finding placement of the outgroup is always easy. It has the most differences or the least amount of similarities in the mitochondrial DNA compared to the other primates. Give me a thumbs up if you're feeling pretty good now with this cladogram. Priscilla's feeling good. Gabby's good. Ava's good. Mare is good. Lots of people are good. Lots of thumbs up. Awesome. Okay, so we're going to let you guys do the next one on your own. So the next one's looking at numerical data again. Be careful because this is looking at amino acid differences. So the numbers are looking for differences, not similarities. And then as you guys do this one on your own, notice that I indicated the nodes. Wherever there's a node, the 2 organisms that are placed on that node, like this organism here and this organism over here, they can be interchanged. It doesn't matter the sequence around a node. Everyone should have the same organism as the outgroup. Everyone should have the same 2 organisms around this node, and everyone should have the same 2 organisms around this node, but the sequence of the organisms within the node does not matter. So you guys are going to do that on your own. And then we're going to collect this data together because next week, I think Thursday of next week, this is when this lab is due, and you're going to build your own cladogram based on the morphology or the anatomy of these organisms. So I'm going to switch out to the Elmo. You guys are seeing my gloves now, right? Okay. So I'll put on my gloves and my goggles, and I have, you guys are humans, you can collect your own data on yourself, right? Like you don't need me to show you a human, but I have the other organisms here. So as we go over these, make sure that you collect the data on them, and then let me know which one do you want to look at first. The crayfish, perch, rat, grasshopper, frog, or the worm? I got them all here.
Crayfish?
Crayfish is first. That's what period 1 chose too. Everybody wants to see the crayfish. Okay.
I just wanted to go in order from left to right.
You just are that type A person. It needs to go a certain way. Okay, so let's look at this crayfish. You can kind of get an idea of how big he is based on my hand. So you can kind of get an idea of his size. That's that little crayfish there. And one of the first things on your table, because we got to go in order, is whether or not it has an exoskeleton. So some organisms have this hard coating, this hard shell, like on the outside. And then other organisms like you and I, we don't have a hard shell. Instead, we have internal bones. So we have an endoskeleton. But this is the exoskeleton or skeleton on the outside. So he definitely does have an exoskeleton. If you were in class, you'd be able to feel this guy and feel that he has a little hard shell. That's in his exoskeleton. So if he has an exoskeleton, he doesn't have a vertebrae because his skeleton's on the outside. And if you've ever caught these or eaten these guys, like they're like little lobsters, there's nothing much, no bones in the middle. But now we're going to move on to bilateral symmetry. So bilateral symmetry means that could I cut this crayfish in half? And if I cut him in half, would it be a mirror image on both of those sections? And I can see eyes on both sides, antenna and pictures. And if I flip him over, yeah, I could cut him in half as well here. So he definitely shows bilateral symmetry. He's a mirror image down the middle. But now we're looking at number of limbs. So I'm going to give you a close-up, and let's see if you guys can guess in the chat how many limbs we're going to say that this crayfish has.
$10?
Yeah, 10. So let's count them. We know he's symmetrical on both sides. We'll count one side. 1, 2, 3. Oh, you know what? He lost a limb over here. Let's count on this side. This is a better side to count. 1, 2, 3, 4, and then this is number 5. The big pincher up here at the top is number 5. So 5 on this side, he's symmetrical. So 5 on the other side. So he has a total of 10 limbs. So we talked a little bit yesterday about the beetles and how the beetles don't have teeth. Instead, they have a mandible, and that mandible cuts through their prey on the outside of their body, and then they bring it inside. So it's a little hard to see, but this crayfish has a mandible as well. And that mandible means that he chews or cuts up his organisms outside of his body and then brings it in, just like the beetle would. So he has no teeth because he has a mandible instead. Obviously, no fur, no hair. What are we thinking about with gills based on what we know about this crayfish's habitat? In the chat, or you can unmute yourself, what would you guess about gills?
I'm guessing yes, like somewhere under the
yeah, it's hard for me to show, but he does have gills for sure. Like he's aquatic, he has gills. And then you can see his antenna are right here. So he has gills, he has antenna. Bipedal means that you walk on 2 legs. He's walking on a lot more than 2 legs. So he's not bipedal. He's walking on a whole bunch of legs. But he does have a mandible. Now when we're looking at body segmentation. So this is a little hard to visualize. It's easier to feel, and unfortunately you can't like touch this guy. And you can kind of see this head, and like I'm trying to see if this head has like a joint in it that would allow this head to move either up and down or back and forth, and it's stationary. It's not moving anywhere. So there's no joint or gap here between the exoskeleton. So this head is attached to this area right here. So this is one big segment. However, notice here, this is flexible. This can move up and down. So I see 1, 2 segments in this crayfish, and that's it, just 2. And then I don't have time to cut these organisms up, but if we did, you would not see a placenta in there. Only mammals have placentas. Anybody miss something on their data table about the crayfish? Okay, Ava chose.
Yes
I'm sorry, Jack, go ahead.
was it yes or no for antenna?
Yes, for antenna.
Yeah.
These are the antenna right here. Ava chose crayfish. Someone else gets to choose the next organism.
Someone should choose worm
Rat.
to spite me.
Rat. I heard rat. I heard rat.
Close enough. Close enough.
Okay. Oh, I gotta zoom out because he's a big rat. He's gross. I'm not a fan of rodents in general, but here's the rat. So you can see how big he is based on like my hand. It's a big boy. It's a big rat. He's a boy. I can kind of see like he has testes there by his tail. But that's our rat right there. So we'll work through this. No exoskeleton in this rat, not like the crayfish at all. But I can feel his bones. I can feel this internal skeleton, and I can feel his vertebra. So I can feel it. So he has that internal skeleton. He has an internal vertebra. What are we thinking about for bilateral symmetry? Does he have that? Could I cut him down his middle, and would it look like a mirror image on both sides?
I would say yes.
Yeah, he does. Yeah, he's bilaterally symmetrical. And then limbs. 1, 2, 3, 4 limbs. Next one is teeth. Okay, let's see. Ooh. Can you see those teeth right there? There, that's better. You can see the bottom teeth then. So he does have teeth, no mandible on that rat, but he has long, disgusting teeth. Ugh. And then definitely, you guys can see he has fur. This was probably white at some time, but he's been preserved dead for a while, so it's kind of yellowed. No gills. He has lungs inside. If I cut him open, I would see lungs. I would not see gills. No antenna. These are just whiskers. They're not antennae. And he's walking on all 4 legs. He's not bipedal. No mandible. No body segmentation. But this is a male rat, obviously, because I can see testes. But if I had a female rat, I could cut her open and I would see a placenta. So we're going to say that rats as a species have placenta, even though this male rat does not based on his biological sex. Anybody miss some data on the disgusting rat? Okay. All right. We have 2 down. We have some to go. Which organism's next? No one's going to choose? Zoe, what do you want to see next? Frog is next. The big frog. So kind of about the size of my hand. So he's a big boy. See how soft he is on the outside? No exoskeleton for this frog. But I can feel the bones in his limbs. Yeah, I can feel the bones. So I can feel the vertebrae as well. So I can definitely feel that in the frog. Bilateral symmetry for sure. Limbs, 1, 2, 3, 4. 4 limbs for our frog. Now, it's the teeth. All right, this is really hard for me to see. And I'm going to try to see if you guys can see it as well. But you see his tongue right there? And I'm looking at the top. I don't see any teeth in this species of frog here. And then his tongue is kind of attached to the bottom. Like I can't even move the tongue because he's a preserved frog. But unless you guys see it, because I know some frogs do, but some frogs don't. I don't see any in this frog. I don't see any teeth here. So I'm saying no to teeth for this frog, even though some species do have them. I just can't see it in this guy. Obviously, no fur, no hair. Gills for this frog, like no, he's an amphibian. He does not have gills. I'm looking over here where gills would normally be on a fish. Yeah, there's no gills here on the frog. No antennae. He is not bipedal because he's going to walk on all 4 limbs. No mandible to chew up food before it enters his body. No segments in his body either, like we would see in that crayfish. And then only mammals are going to have a placenta. We know frogs lay eggs, so there's no internal placenta for that frog because it lays eggs instead. I went through the frog pretty fast. Did anyone miss any data on the frog? Okay, crayfish is done. Rat is done. I need someone to vote for our next organism.
How about the human?
Human, I'm going to let you guys do on your own.
Oh,
because
that would be a dead human.
because I know that you encounter humans all the time and you can collect that data. Jack,
Have
you
you
don't
done
see
human
humans. We don't
cadavers?
see as many humans as we used to, but you guys can look in the mirror for yourselves.
Miss Baxter, have you been in like a lab where you do human cadavers?
Yeah. Mm-hmm.
Oh,
All right, no humans. What's next?
Grasshopper.
Grasshopper. All right, here's our grasshopper right here. We zoom in, it's smaller than our other organisms. So you guys can see him a little bit better. Definitely the first thing that I feel is he has that nice hard exoskeleton. So he has that little outer shell that we're going to see with lots of insects. He definitely has that one too. So if he has an exoskeleton, he's not going to have an internal vertebra. And you guys know that you've probably stepped on lots of grasshoppers in your life and it's all a bunch of goo in the middle, no bones inside. So no vertebrae inside of the grasshopper. However, if I cut him down the middle, he does look pretty symmetrical on both sides. So he does have bilateral symmetry. Let's count his limbs. Big long leg here. That's 1. Here's 2. And here's 3. He's symmetrical, so there's 3 on both sides. So there's 6 limbs on that grasshopper. No teeth, though. And let's see, like it's hard when we're looking at the Elmo camera. If you guys were in class, you may be able to see it a little bit easier. see, if I get too close, then it just gets out of focus. But I can see these external mandible right there. So we have teeth, we chew stuff inside our body, but he's going to chew things outside of his body and then bring it inside. So it's hard to see on the Elmo camera. It's easier in real life, but I see a mandible for sure on the grasshopper. After mandible, what is next? Oh, body segmentation. All right, the last organism that we talked about with body segmentation was the crayfish. And we really looked to see where there was like a break or a joint in that exoskeleton, just look at body segmentation. So I'm going to do the same thing here with this grasshopper. And the first thing that I'm going to do is I'm going to see if this head will move away from that thorax, and we can see, yeah, he's bouncing his head a little bit. So that is, a head is one section of that body. Here's that thorax, and notice that the thorax does allow some movement compared to the abdomen. So head, thorax, abdomen, so 3 sections, 3 body segments there. And then no placenta, it's not a mammal. And then did I skip? I did skip some things. You guys got to yell at me. No teeth, no fur, no gills, but it does have antennae. Obviously, it has 6 limbs on both sides. It's not bipedal. So sorry, I was going too fast with the grasshopper. Did you guys miss anything on your data for him?
Mandible.
Mandible is yes, he does have a mandible. And it's almost easier to see it here without the glare. See it down here? Those little pinchery things? That's the mandible right there. Okay, what organism's next?
Is a perch a bird?
Here's the perch. Zoom out because he's obviously bigger.
Oh, it's a fish.
It's a fish, yeah.
Okay.
There's our perch right there.
Okay.
All right, so we'll go through the perch. So tell like there are scales here, but there's not that strong exoskeleton, so it's a no to an exoskeleton. And then it's hard. Oh, that hurt. It's hard, but like I can't really feel. I can feel it a little bit here. Like it does have some bones. It does have a vertebrae, this perch. You can kind of feel it right here. These hurt. Other things, bilateral symmetry. So you notice I could cut it down the middle. It has the same type of fins on both sides, eyes on both sides, so it is symmetrical. If I cut it down the middle, so it does show bilateral symmetry. Here's when we're going to start talking about limbs. Here's one thing that is a disadvantage when you use morphology or anatomy to do a cladogram. What do you consider a limb? What does somebody else consider a limb? Well, here are some fins. So I see some fins on the sides over here. I see fins on the ventral side. I have this dorsal fin that poked me. So it's up to you if you just decide that those are limbs or not. I'm going to judge your cladogram based on what kind of data that you collect. And if you think these are limbs, great. If you say they're not limbs, that's fine too. So whatever you decide for your cladogram. And then teeth. All right, so let's see if you can, all right, open open up his mouth a little bit and I can see his tongue. So you notice, like, I didn't put my finger in that disgusting rat's mouth, but I'm going to put my finger into the perch's mouth, and what I can feel almost feels like little bristles. It's up to you whether you think that those little like bristles, if that structure is substantial enough for you to determine that to be a tooth or not. But I can feel it. I can feel some bristles in his mouth. And then what else do we have? No fur, no hair.
So are you saying his teeth but not teeth are like humpback whales, how they have those bristly?
He does have bristles, yeah.
Okay.
Yeah, definitely
So
does have bristles.
it's not like a piranha where they're like really tiny.
No, it's not like that, because if it was a piranha, I definitely wouldn't be sticking my finger in there. Like, that would hurt. But the little bristles are there, but they don't hurt me. But like if I was smaller, I'm sure they would. And then do you guys see his gills? Like, that's easier to see, is those gills there. So definitely gills. No antenna. He's not walking anywhere. He's not bipedal. No mandible. No body segmentation. He's not a mammal. He does not have a placenta. Okay, so we just have the worm left, right? Here's our worm. A whole lot of nothing to this worm. No exoskeleton. He's super soft. No vertebrae at all. Everything soft about this worm. But he does have bilateral symmetry. So I could cut this worm in half and I would have a mirror image on both sides. And even if I cut through them and I looked at the organs inside, I would see some bilateral symmetry in that worm. Obviously, no limbs on this worm. No teeth, no fur, no hair, no gills, no antenna, no legs, so it's not bipedal, no mandible. But do you see all of the lines? Every single line is a segment here. So these are body segmentation. Many, many, many segments in this worm. And then no placenta for him. So the only really data on the worm is that you know that he's symmetrical and that he has lots of body segments. Other than that, you have a whole bunch of minuses for him. As you guys look through your data table, is there anything that you're unsure about? Okay, the only assignment that is due this weekend is the human skin color assignment, and that's due tonight at midnight. However, we have a 3-day weekend. So if you want, you can progress through this cladogram assignment, but it's not due until next Thursday. So look at your calendar. Do you have a lot of time? Maybe it's good for you to kind of work on this and get this done now. Or do you need the weekend to rest and recoup? If you need that, that's fine too, because this cladogram assignment is not due until next Thursday. But when you do make your cladogram, you're making it from scratch. Like, I didn't give you like a skeleton for it. I'm going to make it based off of your data that you collected and 7 traits. We collected more than 7 traits. You can choose which 7 you want. If you want to do all of these traits, that's fine. But you only have to have 7 traits on your cladogram. And then I see one question in the chat about the limbs of the grasshopper. I see 3 on both sides, so that would mean 6 limbs in total for the grasshopper. Well, any other questions on this? Okay, guys, I will not see you, remember, until Wednesday. It is a long time till I see you again, so I hope you have a really good weekend. Reach out to me if you need me about anything, though, and